So we're talking about our efforts to develop gene therapy for people with blinding diseases owing to gene defects. So for the last 20 years or so, we've been working to develop new treatments for these conditions which have currently been essentially untreatable. And we're excited to discover that by providing the retina with genes that they lack, we can actually improve the outcome for people. 10 years ago, we performed at Moorfields the world's first gene therapy for inherited eye disease. And last year was the first treatment made available, approved and licensed for use, albeit for one specific rare condition in the United States. We very much hope and expect that many more treatments will be available, not only to rare conditions, but also to more common conditions such as macular degeneration, glaucoma and diabetes, using elements of the technology that's been developed here at Moorfields and elsewhere over that period of time. We've seen significant improvements in the ability of people to see in various aspects of their sight. So specifically, children with a particular form of inherited blindness can see better in dim light. We're very much hoping that that'll translate into an improvement in terms of their ability to see better in the future. So Mark is one of many individuals, many children who've benefited from this approach. It's important to be clear, this is a very rare genetic disease, but we are very optimistic that with further work, further development, there will be more opportunities for development of treatments like this for other children and adults with inherited retinal disease and other more common conditions, again, as I mentioned earlier, like macular degeneration, diabetes and glaucoma. I think there's real optimism that these kind of techniques will be useful, will be valuable for specific forms of inherited blindness, including some forms of Leber congenital amaurosis and RP. At the moment, we are optimizing it so as to identify which forms are most likely to benefit and to optimize the technology in order to provide similar treatments for other rare conditions. It's now possible to provide cells with healthy copies of the gene that they otherwise lack. And these copies can be packaged a bit like in a capsule, but actually we use modified viruses. Viruses can be modified to enable them to deliver genes into the cell quite safely, because viruses are highly evolved to do exactly that. And we can get millions of the genes, package them in millions of viruses, and inject them into the retina in order to provide the cells that are lacking a gene with it. And that's exactly what we do. What we're seeing now is the very first relatively simple interventions to target gene defects that are recessive owing to a loss of function. I think that this is only the beginning, and in the future we can expect similar techniques to use genetic manipulation to manipulate cells, the genes inside cells for a variety of different conditions, including glaucoma and macular degeneration.